What is going on, everybody? Welcome to another awesome show here on Logical, Plausible, Probable. Today, you and I will have the opportunity to learn about some really incredible discoveries that have been made over this past decade. And we're compiled into a theory called the MESHcode. And we are going to be able to gain this knowledge directly from the man who published this new theory himself, Dr. Ben Goult. He's sharing his time with us, and it's going to be a very fun conversation. And I tell you what, folks, I always like to give a little preface so that anybody who has questions about, is what you're about to hear true, false, or made up, what's going on? So Dr. Goult has a pretty interesting background, and he's a structural mechanobiologist. That's an awesome concept there, who combines structural biology, biochemistry, biophysics, and mechanobiology to define how physical and mechanical forces are sensed through extracellular matrix adhesion complexes. Wrap your mind around that, and how this controls cellular processes. He has developed an international reputation for his work on the protein Talin, and actually has his own research group that has defined Talin as a major mechanosensitive signaling hub, and that it has molecular memory, which enables organisms to store data through persistent alteration of protein conformation. Dr. Goult, thank you so much for your time. I know you're getting ready to go for a trip, so thank you for sharing your time with us and educate us on your amazing discovery.
No, it's great to be here. Thanks, John. And thanks for the nice introduction too.
Oh, it's well justified. And like I said, I always find it interesting how folks like yourself will take the time out of your busy schedules to share your knowledge. And I think very often, especially in the modern world, I find it very fascinating how we have so much access to information, but so few people are aware of the innovation and the discoveries and all these different things that folks like yourself are being able to discover on a rapidly accelerating level. I think. I don't know what your thoughts on that are, but it seems like we're just discovering new things on an almost daily basis that having a hard time keeping up with everything. Is that a fair subject to somebody who's in the research field?
Yes, it's moving frighteningly fast. This last 5 or 10 years, it's just and all the new technology, all the new ideas, it's all converging to a greater knowledge, definitely.
Now, for my audience, you guys know that I'm very fascinated by biological codes, the nanotech, the computation that's happening on the molecular level, and obviously its implications to biotechnologies and all the things that we're doing in human innovation and trying to hybridize our technologies. But Dr. Goult, can you please kind of give us an overview of the incredible implications and potential of your discovery? And can you guys give the audience, what is MESHcode? What's the general concept of it, and a bit of a backstory for how you realize what's going on? Because I know this has been, I think you were working on this since about 2007, I think, 2008, somewhere around there.
Yeah, 2006. I started working on the protein Talin, probably 2005 indirectly. So I've been working on this for a long time. And originally we were just very interested in how cells can migrate, how they can crawl around, and the machinery which they use to do that. And it's via these adhesions which the cell makes. So we're very interested in understand how the cell knows where to grab on, and how it grabs on, and how it can coordinate that process. And how it grabs on is that all of our cells sat on this layer, this protein meshwork called the extracellular matrix. And it surrounds all of our cells, and our cells grab onto this, and it gives them instructions on what to behave and how to know that they're in a kidney or a lung or the brain. It tells them instructions. But how this happens, and how does the cell detect that, and how does it hold on? And the main way it does this is by a family of proteins called integrins. And these are like hands of the cell which physically grab onto the extracellular matrix. And that's really interesting in its own right. But they're on the inside of the cell, just inside the cell, there's these large complexes called adhesions. Focal adhesions is the common name. And the main part of that is this protein called Talin. And that's what we're going to talk about today. And that's what my focus of the research has been on. And Talin links these integrins and these hands of the cell and the cargo net which they're grabbing onto. It links it to the cell's fourth generation machinery. This is called a cytoskeleton. And that's a load of motor proteins which can push and pull, and a load of filaments which it can grab onto. So that's a bit of a convoluted way, but we're very interested in how Talin could make this linkage. But then what we got, as we started to learn more and more, what we started to realize was that these cells, not only are they grabbing on, but that's leading to a total reprogramming of the cell depending on what it's attached to. So there's some sort of computation aspect. And it was trying to understand that, which just led to where we are now, and led to what the MESHcode theory is. Should I go on a bit longer?
No. You're that's very interesting. So you are you suggesting that one of the things that you guys kind of recognize was that we have the equivalent of like if-thens happening that are somehow being accounted for, and we've got to figure out what is the we're observing these things happening, and we've got to figure out what is the kind of the cause behind it. Is that?
Yes, possibly. Yeah, and I think the best way to show that this is information processing which is going on in these adhesion sites was an experiment from Dennis Disher's lab about 15 years ago, where they took some stem cells, and these are a type of cell which is undifferentiated, so it can differentiate into any type of other cell, and it can go into like a fat cell or a brain cell or a muscle cell or a bone cell. And the experiment they did was to keep everything constant, apart from just change how stiff that extracellular matrix was. And if they made it really, really soft, then those cells would read that, grab onto that, and become a fat cell or a brain cell. If they made it like really solid, like glass, then they would reprogram into a bone cell. So somehow that cell was feeling its environment, and then the instructions from the physical properties of that environment, how stiff it was, how hard it was to warp and bend, it was grabbing on and then converting that into which genes were switched on and off. So there was a clear information processing step happening at these adhesion sites, which led to the whole cell reprogramming and doing a different role. And that was what we wanted to understand, how that happened.
So sidebar question. Does this mean there's a potential implication of what we'll talk about here as we go throughout the interview, but is one of the potential implications of what you're researching now, could it potentially be able to, if you can figure out what is controlling these signals, you could potentially convert a cell back into a stem cell? Is that a potential end outcome, or modify it away from what it is to something different? Or because I know there's some research going on into that into that premise. Is that something that you guys even considered or is being looked at, or is that a completely different context?
No, that's kind of, and a lot of people are doing that by changing the surface or the patterning, the shapes of the surfaces which they can grab onto, and in conjunction with some signal factors like they can undifferentiate cells to a certain level. And there's a whole field of research in this alteration of cells back from being differentiated back into being stem-like. So yes, I think, and in part of that is the mechanical and the geometric patterns of what they're attached to.
Interesting. Well, okay, so you know, a lot of people are told that the processes you're referring to and all these adhesions, all these different things are just, you know, in a biological system, that's just a chemical reaction, and they're, you know, very unaware of, you know, the word you've been using, mechanical actions that are occurring to, you know, result in these end outcomes. Can you kind of touch on that for a little, for a second, and then also explain what the concept or the term mechanosensitive signaling hub? Can you kind of give us a little overview of what that means as well?
So all of this came because as we were doing more and more experiments on Talin, and as more and more things it was interacting with and jobs it was doing, we were trying to understand how they happen. And the big step change in the field of research I work on was we were working on two binding partners of Talin, so two proteins which interact with a Talin. And we mapped where on the Talin molecule they interact, and they both bind to the same region, the same domain, but they bind to the domain in two different shapes. So it leads to this idea that there's some switches, because this domain is like a helical bundle. So it's got like, it's compact and it's closed, and it binds to this protein called RIAM in that state where it sits on the edge. Maybe that way. Have you got the figure, actually, the figure 1 of this? No, figure 2. It might be easier to share. In fact, I can share mine. Oh, you share. Yeah, this. So this is what the Talin molecule looks like. It's got lots and lots of these domains. And for a long time, we not me, because it's so well conserved, but a lot of people in the field said that this was just a spacer. This was just a linker to connect the integrins and the ECM at one end, the matrix at one end, to the cytoskeleton. But each of these domains labeled R1, R2, R3, these are these domains, and these are switches. And what we discovered was working on this third one, this R3, and that was because it does one job when it's in this closed state, and it does a completely different job when it's in this open state. And that immediately showed a way that you could read physical properties of the surroundings and of the force generation. Because if you cause that switch to change from a closed state doing one job and one set of signals, open it up to do a completely different job, then if you open and close that domain using force, then you can switch on and off different signaling pathways. So it's not that this is just a random chemical reaction. It's like these binding sites are either there or not, depending on the mechanical signals. And where that led to this notion of a mechanosensitive signaling hub was that here I'm just talking about this one switch, this R3 going from an open and closed. All 13 of these have the same switch-like property. So on this domain, there's 13 binary switches in a row, which I try and show conceptually on this right-hand side, because you can start to imagine that these domains can be in this folded O state or open up to this unfolded 1 state. So you can push and pull on these molecules different numbers of times and recruit lots of different things to do different jobs. Do you want to come back in? I could talk all day, like, but that's why it's a signaling hub, because depending on the mechanical environment, depending on how stiff it is outside, and depending on how much the motors inside are switched on and off, those different parameters result in specific patterns of folded and unfolded domains, which then switch on and off different signaling pathways. So the whole thing gets it couples mechanics and physical environment with signaling pathways to give a mechanosensitive signaling hub.
That's very interesting. So you're saying that each one of these is R1, 2, 3, etc. is all operating as basically like almost like logic gates to an extent to represent the 0 and 1 on off switch and then enable other pathways to be executed based on whichever one is open dynamically. Is that correct?
Yes, pretty much. So once we found that R3 was doing these two different jobs when it was open or closed, one of the things we were really keen to do was to understand how hard would you have to pull to make this switch open, and whether it would close straight away, or what would cause it to close. So we set up a really long-standing collaboration with, in Singapore, there's a Mechanobiology Institute, and there we can take a single Talin molecule and we can manipulate it using a magnetic tweezers, it's called. So we can grab a molecule and then we can apply force and pull on it, and then we can watch these switches switch and see how they behave, how much force do you need to make these switches. And it's quite striking because it turns out that that first switch in Talin is the force generated by the motor protein. So you can pull and flick the switch and set the whole thing into action.
So in line with that, and you and I kind of talked about this off camera. I know you mentioned this in your paper as well. There's I know it was well over, I think you said almost close to 250 different proteins that are involved in this. Are you saying that obviously in your experiment you were using the magnetic tweezers you were talking about?
Yes.
In the biological system, are there proteins that are executing that exact amount of force and able to open and close these switches?
Yes. So that's the idea, that as the cell's moving, or when it switches motor proteins on and it's generating contractility and pulling on these switches, as they flick, will change the signaling and then either turn up or down contractility. So basically the whole thing works. You pull on it and then you change the signaling, and then it either makes it pull harder or less hard. But there's this like these interdependent feedback loops where the pull causes a change, which changes the signaling, which then might make another pull or might alter the whole thing. But ultimately, up to 250 different proteins will come together in different combinations, depending on these physical parameters. So basically, the cell's using physics in a really interesting way, and that's what we want to understand.
Interesting. So all of this, right now we're talking, for the audience, right now we're talking about the mechanics of the physical actions that are happening in order for this, but the ramifications of all of this is that and this is where the mesh code really starts to come into play, is that there's actual data and information being stored through these processes. Is that right?
Yes. And the realization comes from the experiments where we were pulling and put on these switches, is that they take, you pull with a certain amount of force. Say it takes 5 piconewtons of force to unfold. The realization came that if you take 5 piconewtons, you'll unfold it. But if the force goes down to 4 piconewtons, it won't immediately refold. It will stay in the open state. So they've got this, it's called hysteresis, where the force to unfold it is much higher than the force at which it will refold. So when you pull it into a state, and it will stay in that state until the force goes back down quite low. So they store this information. So what we start to imagine is this Talin molecule can have lots of different shapes as a function of how many times it's been pushed and pulled. And they will store that information for quite a long, well, if you're in a perfect mechanical system, it would store that information permanently, but there's no such thing as a perfect mechanical system. Where the memory comes from is you've got this hysteresis where they stay, they have that short-term memory, but on top of that, you assemble this 250 different proteins, which sort of fix those patterns by making big complexes. So it's the memory comes from the things which interact with it. But the idea is that these patterns of ones and O's basically depend on the history of events which have happened to that molecule. So that's carrying information forward in time, because depending on the shape that molecule's in, when you pull on it again, you'll get a certain pattern depending on what shape it was already in. And that's the general premise beneath the mesh code, is that these are memory molecules that we're working on. And it took me 15 years to realize that these were memory molecules, but this information is persistent patterns of binary strings of information.
Right. Okay. So in line with that, so for the audience, neuroscientists have been, mapping out different regions of the brain and presenting all these different hypotheses over that, a long time, especially over this last 15 years, right? And all the types of actions that are being controlled by what portion of the brain. But correct me if I'm wrong, but how memory has actually been stored has been a rather elusive concept. Hey, we know this type of memory is going here, it seems like, but we don't necessarily know how. Are you suggesting that mesh code could represent, I would use the word binary, but could represent a digital memory or digital mechanism for how memory is being stored by organisms? Is that kind of what's being implied here?
Yes. So again, with that stem cell reprogramming, these patterns fix, but then over time they can lead to total reprogramming of the cell. They can switch on different genes, different epigenetics, but they can capture that information in the synapse. So another key thing to mention in this, I don't know if you've got my figure, I don't know, number 2, to show where these switches are in terms of maybe the one before, maybe the next. I don't know which one. Yeah, this one. So this is supposed to show what a synapse looks like, where you have two nerve cells which come together, and they make a tight junction like this, where they could send a signal from one, and it will signal to the other. So these are basically cell signaling centers, where you can send a really discrete signal between two nerve cells. These aren't just stuck together like this by themselves. They're held together via an extensive scaffolding, which holds them in this shape. And that scaffolding contains a lot of extracellular matrix and a lot of integrins. And talin's an essential part of that scaffolding. So what the idea here is that this scaffolding, which holds these synapses together, has got these switches in. So we know these memory molecules exist because we can show that experimentally. And here the idea is that these memory molecules, which are shown to be part of the synapses, the idea is that when the signal, the electrochemical signaling, is going around the brain, it would be causing contractility in these synapses. Again, that contractility is appreciated and has been shown experimentally. But the bit we're putting here is that contractility, when it pulls, would change these patterns of folded and unfolded, patterns of ones and zeros in that synapse. And in doing so, it would encode information into the scaffolding, into the shape of the scaffolding molecules. And then that would then affect the activity of that synapse. So you could send information to different parts of the brain. And the electrical chemical signaling would be constantly updating these switch patterns.
So one of the implications of this, I'm going to ask you this question a little later on in relation to, you make the allusion or the direct comparison to SSD storage, but are you suggesting that this is indicating dynamic read-write capabilities that are being executed here?
Yes, that's definitely what I'm suggesting. That's quite speculative at the minute, but what we can do read-write on a molecule. We can coordinate that, and we can write information, and we can read that using probes to read the different shapes. So the idea would be, yes, that you could physically write information into each synapse. Because I don't know if you've got my figure 6 in this thing, but the last figure in that shows the similarities between maybe I can share it, if not.
Yeah, I got it right here. This one?
Yeah, so this is what the cortex of our brain looks like. This is a really old, I love this figure. It's from 86, I think, or 96. But it's just showing 3 of these what's called cortical columns. And there's 2 million of these in the brain. But the thing which really leaps out in the context of, so basically what I'm saying here is that there's a physical location for information written into the scaffolding of the synapses. So there's a physical way to write information into matter. But what I'm showing here is this incredibly repetitive nature, where these red teardrops at the top in this layer 2 and 3 and IVA, like tier 4, each one of these is a pyramidal neuron, and they've got up to 60,000 synapses, and each synapse has got we're trying to get hard numbers on it, 100 talins, which have each got 13 switches. So this represents, in my opinion, and what I'm trying to propose, and would love to be able to test, is that this is a vast array of memory molecules structured in a really high ordered way. And then on the left, this is what the architecture of a SSD card looks like, where you've got this incredibly repetitive logical structure of many different channels, each one subdivided into many different NAND flash, they're called, which have got pages and blocks, and then the actual ones and zeros. But it's the same, it looks like the same kind of architecture broadly, that you've got this repetitive thing all feeding back to a memory module, a memory controller, to send information to specific sectors.
That's interesting.
I mean, it's absolutely, it's a bit like, sounds a bit wacky really, but it's based on solid experimental data. We can, on a single molecule, we've got this. Here we've just got what I'm proposing is we've got a vast array of these memory molecules ordered in a higher order than what we'd currently imagine.
Right. Well, and let's oh, get an echo there. What's happening here? Let me see. Let me mute you for a second. Real quick, I'm getting an echo. Let me see if I can fix that. So one of the points, and kind of line what we're talking about now, one of the points you make in your paper, and by the way, folks, the paper is linked in the description. I highly recommend you going and reading it. It's very fascinating stuff. And one of the points you make is discovery. Your discovery is indicating, and you're as we're referring to now, that the brain is operating like a molecular computer, or sorry, a mechanical computer on the molecular level. And I want to quote from your paper and then ask you to kind of expand on this as we've kind of been touching here. You say, quote, "There are considerable similarities between a mechanical computer and a cell. Each cell contains a series of levers, pulleys, and gears in the form of its cytoskeleton. The cytoskeleton is an incredibly complex dynamic network formed of three major classes of filaments: actin, microtubules, and intermediate filaments. There are hundreds of cytoskeleton regulators that control these networks with the precise linkages, filaments, and adapters determined by the programming that cell is running. The adhesions to the ECM, mediated by the integrin family of ECM receptors, serve as information processing centers, able to feel the surrounding environment and instruct the cell how to function." Now, you kind of alluded to this earlier when we were talking about the different ways that cells were formed, the stem cells, those kind of things. Is everything that we've been talking about now? Can you get into the intricacies a little bit of the, why you're calling it a molecular computer? You're kind of alluding to this and showing with the allusion to the NANDs and everything else, but a lot of people don't want to really accept the premise that there's the direct equivalence of computation and a mechanical computer executing processes here. Can you kind of dive into that a little bit more detail?
Yeah, I can try. So the general premise there is that the cytoskeleton, this network of filaments connected to motor proteins, connected to mechanical switches, provides a way for the whole cell to work as a computer. Because if you stimulate the motors at one end of the cell, it pushing and pulling and trying to work it out. So the idea is that that's like you're switching on the computer and it calculates and it balances out, and it's the balancing out of the whole system, which is the computation. So it's not necessarily like a computer like we'd imagine where we turn it on, it works, but this thing gets turned on by a signal, like a signal comes in, turns on some motors, they push and pull, which propagates across the whole cell. And then that, when it gets to the end state where it's finished, because the whole thing is you don't want the engines whirring the whole time, so it tries to reach a balanced state. So I think this is like an energy minimization where all of this factors balance out, and at the end you're in a certain pattern where the patterns and the shapes of the molecules are in a meaningful arrangement.
The point you just made about the energy reduction aspect, what struck me when you said that was, I remember reading some stuff a while back about how one of the things that blows people's minds about the brain capacity is the energy requirement being so low. Yes. And that if the equivalent amount of computation being done by our human created computers, then the energy demand would just be absolutely astronomical. Is what you just said about what you're observing, could that have some relevance to the point I was just making there about the energy demand? Yeah,
because I think that the whole cell isn't constantly pushing and pulling the whole time. It's trying to get to a state where it's held together, like the whole thing is balanced, but it's like under tension. Like if you're in a tug of war, you can either both be pulling absolutely as hard as you can, or you can just hold it at enough tension so that the thing is maintained, but you're not using a huge amount of energy. And I think it's like that. I think you want to balance this out, so you're using the least amount of energy, but still maintaining the connections and the network and the linkages. And that's what I think happens, because just inside these synapses, and what's really well established is inside the synapse, there's lots and lots of these things called gaps and gefs, which are basically, in the simplest sense, they switch on and off the motor proteins. So when you have a signal going across, it temporarily switches on the motors, which then starts this calculation. But the default is to get back to a low energy state, because as you say, you'd burn through your glucose in high speed if you were constantly pushing and pulling, or if you was constantly sending, because some of the theories are like it's all almost held in RAM by these electrical chemical signaling constantly going round, but that would require huge amounts of energy. Whereas here, a low force state is actually better for preserving this mechanical state. So these switches behave better if you keep them at a low force, but then give temporary spikes of high force. They'll store the information much better.
So one of the things you and I kind of talked about before we started was the the implications of the specificity of all of this versus just being random, uncontrolled reactions and such. Is with what you're talking about now, that seems to lend a lot of credence to that point, that it's not as nearly as chaotic and random as some people like to talk about, or it goes kind of previous theories of how systems work. Oh, it's just a bunch of more random reactions that ultimately end up somehow in this end outcome versus a very specified, controlled execution of a variety of functions. That is what I'm saying. Would you agree with that? Disagree, or kind of somewhere in between? What are your
I would
thoughts
say
on that?
that if you look at a cell with a microscope, with a fluorescence microscope, and just look at it in a culture, in a dish, it looks incredibly dynamic. Everything's going, all the motors are going, it's sending out protrusions, everything's turning over really fast. So it looks like it's chaos. Well, not chaos, it looks like it's order, but a lot chaotic. That's because that cell never reaches equilibrium, because it's not touching its right neighbors. It's not making the right patterns on the surface, getting the right signals. So it's constantly trying to it can't complete its minimization. I don't know if you've ever seen the Babbage analytical engine, these computers, which you turn the wheel and it starts doing it.
Yeah,
If you
like,
imagine,
1837, I think, something like that. It was
yeah, so I think
very
at the Science Museum in London, they rebuilt it, and they basically with his old blueprints, because I don't think he ever quite got it to work. But if you imagine that, you're turning the wheel and it's doing the calculation, it will only reach a solution once it all comes into place. So if you were to take one of those cogs out and drop it into the bottom, then it would just keep going and going and going, because there'd be no solution to that problem. And that's what you're seeing when you look at cells in culture. They're constantly trying to do stuff. So they look like there's order, but you're only getting glimpses of it. Whereas what I think is actually happening is that in the context of an organism, then all of that collapses into an ordered state. So everything becomes ordered. And that's really controversial at the minute, but it's an extension of where the concept of order, that's what we're looking at, ordering these molecules, the shape of the molecules as a way to encode information. So I think there's a higher order of stuff than what we currently appreciate.
You know, the point you're making about what we quote unquote see, right? I was watching a lecture. I'm drawing a blank on the gentleman's name. He's at Stanford, I believe, but he was talking. The lecture he was giving was on the microtubule pathways, and how some of them are one way, some of them are two way, some of them only go to go out to the membrane. It was very fascinating. They were showing, and one of the things, the points he was making was, as our imaging technology continues to increase, we keep finding these more and more amazing different controlled functions that you're in this context, it was microtubules that this one, and they'd figured out how to reprogram them to this one that normally was only in this direction. They'd figured out how to make the switches, and now it could be a two-way or go the opposite direction, those kind of things. And I think, correct me if I'm wrong with this, but I think kind of what you're alluding to is, as our imaging technology continues to improve, then the probability is we will find more and more intricately controlled systems rather than just random chaos. Is that a fair point?
Yes, but the imaging needs to, like, I think we would already be able to see the order if the cells were ordered. I mean, it's actually the limitation of how we do the experiments of trying to visualize cells in a microscope. So as we can get more intravital imaging and stuff, we might get more glimpses of this. The thing I could tell is that you try to lead towards this ordered system, but the cool thing is that these ordered systems come from is in essence 3 or 4 different proteins which interact in different ways depending on physical parameters. And then that provides a scaffold which coordinates a lot more processes on top of that. So it's a layer on top of it, but the underlying layer of where you could get all of the different, a lot of the different architectures, and could be with quite a few basic building blocks, and then you could layer and layer on complexity. And I think that's one of the interesting things for me is, like, what would you take to actually begin to build these systems in vitro, in a test tube or in a petri dish, like what would it require, and to try and build it up from that level.
Here's a random question. Are there any, and if you may or may not know this, are there any known like mental problems that are associated with like mutations or errors in the Talin proteins, or any of the ones that are associated with it in terms of, cognitive function?
There's very few mutations associated with Talin in general, because it's so essential, because it's the core of these complexes. So any bit which goes wrong, it's not. There is a couple which aren't published yet, which we're working through, but the proteins which interact with this and sit on top, the next layer of information. So I think these switches at the bottom, they're essential. If anything goes wrong with that, then you're not going to be viable as an organism. And we can see that, because if we put a mutation in, then you can't, like a fly, they wouldn't like it. So they're so fundamental. But the things which interact with the switches, there's loads of diseases linked to those, and there's lots of, and the things which switch on and off the motor proteins themselves, then they're linked to a lot of learning difficulties. And so yes, the mechanical linkages are really important.
So what you're saying is, basically, if you have any errors in the Talin proteins, you're probably going to be your body, the organism might terminate the reproduction if it's detected, kind of thing. Or it's not going to be viable.
Like there'll be some stage where one of those switches or one of those things needs to be working at the right way, and then it'll fall down, because it looks like development's a string of events. Once it starts, every step is dependent on the previous step being almost at the right state. There's a lot of scope for tolerating imperfection, but by and large, one step follows the other,
right.
And one of those steps will fall down. So there's not really any mutations in Talin is the short answer.
Interesting. Yeah.
Which have been discovered yet. Now it's really cheap to sequence people. We're probably going to find a lot more, because if you present with an illness or a condition, then they'll sequence you and they'll say, here's a mutation in your Talin or whatever. So more and more information is coming online all the time, because when they did the human genome, it cost billions and billions, probably £1 trillion to sequence. But now you can get a human exome sequence for under £1,000. So it's come right down. So if you've got private health insurance, then they'll sequence you just as part of your diagnosis, I reckon. So,
right. No, that's very interesting. My curiosity is piqued on all of this. I'm wondering if there's going to be implications. The point you're making about being able to do the comparative sequencing, I'm wondering if there'll be correlations between IQ and level of cognitive function and the overall efficiency of not just the Talin, but also the other proteins you're referring to, all working together. I wonder if there's any ramifications or implications in relation to the overall efficiency of cognitive function. Is that anything that's being considered?
I don't know. I think mechanical computation, that's the idea I'm trying to develop, is like this mechanical computation which occurs. There might be different amounts of efficiency of that, and maybe that correlates to certain diseases. But I think it's a long way. There's a lot of complexity on top of this, because what I'm talking about here is stripping this down to its absolute bare bones, and then on top of this, there's a lot of stuff. So I wouldn't feel like, no, it's not something we're considering to try.
So it's kind of like saying, this DNA sequence equals X protein, and being like, that's it. In terms of the processes that are involved in just getting from A to B, they'd be like saying, oh yeah, there's a Talin protein, therefore we've explained everything. Which obviously is not even close to being scratching the surface.
Memory, and we understand cognition, we're nowhere near. But something which is quite interesting is that a lot of the genetic linkages for things like Alzheimer's, they correlate with adhesion proteins, so not with Talin itself, but with the things which control Talin. So these adhesive structures, and by extension, these patterns of ones and zeros. Okay, for you guys over there, I think O's is not. This binary coding, or this digital coding, is tightly connected to the genome-wide association studies of Alzheimer's. So it might be that the Alzheimer's disease and some of these diseases are corrupting this information, because if it was like an SSD card where these ones and O's contain meaningful information, and these synapses have got specific things, then if that pattern gets corrupted in any way, then that will no longer make sense with the rest of the interpretation of it. So if like you scratch a CD, then that bit, you can no longer get that information back. So if you're playing your favorite song and it's got a scratch on it, you'll always get a bit of noise at that bit. So it might be the same thing where if you start to corrupt these patterns, it's no longer a meaningful thing. So when it tries to rebuild that thought or that memory, it might not be able to do it.
So that could have implications in the point of, like with someone with Alzheimer's or other forms of dementia, they have portions of their memory erased, and now they're viewing something from their past as having happened from their perspective. It happened yesterday, kind of thing, because you've deleted or damaged the memory in between those stages. That or maybe they remember a portion of an experience, but not the totality, and it's like it's been that one portion's been erased, but they remember this, the first segment and the last segment, but they've missed the middle, or you forget the beginning, the end, that kind of thing. Is that where you're
and it might be like the different, because it's not like the switches are probably just one way to capture this information, but maybe in the long-term storage these become modified so they're locked in certain patterns, or the whole cell might reapproach. So there might be like a really undynamic thing. So the stuff we remember from being a kid might be quite locked, like long-term. And the bit which is constantly being done on the day-to-day might be okay, but the bit in the middle where it's more transient memories before they get fully written, if that bit is flaky because of this damage or because of this missignaling, then that might be why it doesn't all quite connect. So your whole story no longer makes full sense. But again, it's quite speculative. But the idea we'd like to try and think is maybe if it is screwing up these patterns of ones and zeros, then maybe there's a way to stop that bit prior to when you start to see these big plaques in the brain. I think most of the effect has already happened long before. And I think that's being borne out by a lot of these failed Alzheimer's drugs, which are coming online all the time now or being abandoned, because the plaques probably aren't the actual causative thing. It's probably the disruption of this information at an earlier stage. So that would be where we'd be looking if we were going to try and get at this. And that's one of the things we were looking at in the lab, is to see, could we understand the interplay between the amyloid proteins and adhesion and try and understand that.
Interesting. Okay, so yeah, I follow exactly what you're saying on the plaque. I mean, obviously if you read the general literature for the public, right, on WebMD and all that kind of stuff, it's plaque building up and is the cause of all that kind of stuff. And if I'm correct, what you're saying is that may have some kind of implication at some level, but we're not even addressing the root cause. Is that?
There might be a really much more toxic intermediate on the way to a plaque, which
that might be the end outcome, but not the root cause.
It might be that the brain just puts all this stuff out the way in these lumps to try and protect itself. But the bit which is scrambling the information might be a extra reactive or extra species which is formed at some point, and that's what's causing the damage. So that's what we'd be interested to look at. But again, it's speculation, but it builds on this notion that if you look where this amyloid precursor protein is in a cell, it's at the adhesion site. So it leads to this notion that, anyhow, that's a bit down the line. Now, I'll talk to you about that when we work that one out. But
no, that's very cool. Now, okay, so let's you and I've been going down some rabbit holes. I'm fascinated by all this stuff. Let's take it back to a concept that is fundamental to what we're discussing right now, but I think we want to make sure the audience kind of grasp here. So throughout your paper, you used the term machine code, but one of the sections that really struck me was I'm about to read, and I'd love for you to expound on this point here. So furthermore, this quote, "Furthermore, mesh codes are found in all places where cells engage the ECM. So having a machine code running in every cell in the organism would hint at a unifying theory of cell communication, which provides an instruction set for each cell to work in synchrony. Like computer machine code, without the necessary parsers to correctly decode the symbols, the code is unreadable to an outsider and would look like it's just pulses of electric activity, triggering alterations to vast strings of ones and zeros. However, once the language of the code is understood, this information might be decipherable and reveal an unimaginable level of communication." Can you kind of expound on that? And I've got some follow-up questions, but just kind of give us the general overview of what you mean by the implications of what we're talking about.
Yeah, so basically exactly what it says there. I think that was actually quite succinct to me to write that. I'd probably say a lot longer to describe it. But basically, all of the cells in our body are making these adhesive structures, and this is holding them in the right place. But because of this like with the stem cell experiment, this process called mechanotransduction, where the mechanical state of the cell is talking to the genome and controlling the which genes are being read and which ones are shut down. That these patterns of information on the bottom of the cell might be constantly be able to be updatable, which can then lead to ordered changes in what genes are read, what splicing occurs, all of that type of stuff. And in which case, then that means that this cell would be talking to this cell, which would be talking to and they would. It might be that the cells are sharing this information, and that like the computation within each cell is like having two cells. You might get 10, like they can stack up and like lead to all of the cells are talking and updating each other as a function, and it's like parallel processing of all of this computation. And in which case, if you strip it right down, which I am doing, this is massively oversimplified, obviously, but at the core, this would be patterns in these switches of ones and zeros in each cell, and then signals would come in and it could change those patterns. And it might just be that if every cell is running the same mechanical coding and the same genetic coding, then there's the possibility that this could be a machine coding, because you could send a signal to update the mechanical coding, which would then lead to the changes. So they'd all be working together.
Well, and the point you made about the the necessary parsers to correctly decode the symbols.
Yeah.
That obviously for the general audience, excuse me, a basic concept of this is, you have codons in an mRNA sequence that are being, translated by the ribosomes, and, tRNAs or there are multiple components all working together to read the information there. You're suggesting that there's other types of decoders involved in the extracellular matrix that would be translating the signaling and transmitting the sender and receiver aspects of it. And we basically need to figure out the point you made in that section was the implications of the language. Hey, if we can figure out what it is, then we'll understand what's going on to control it, obviously. But are you saying that we figured out the syntax of the genetic code, now we're trying to figure out what the syntax is of the mesh code? Is that
yeah, and I think so. We kind of know what the syntax is of these mechanical switches and this mechanical computation, but it's how they connect together and how one cell's status is updating the other cell, so they all synchronize relative to each other. That's basically what I'm trying to say, I think, in a better way, is that all the cells in our body synchronize to be part of the same organism. We're not like just a big pile of amoebas on the floor. They're all attached and they're all synchronized in this way. And it's the level, and in terms of like the electrical activity, I think there I'm more alluding to like brain activity, where you put an EEG hat on and you measure the activity, and it just looks like loads and loads of electricity going round, and in nervous impulse and all of that. But that would be actually be updating an underlying code underneath that, and then that would be leading to the effects downstream. So that would be the idea.
Interesting. So,
but it's the synchronization of all of the cells relative to each other, which is the interesting bit.
Gotcha. So you're saying, okay, we've kind of figured out the syntax. Now, are you attempting to figure out the semantics? I guess would be the next stage of this pattern equals X outcome. Is that where things are? To me, that seems like a very, very deep, dark hole to be going down, or trying in a good way of trying to figure out what the heck all this means. Because if we can understand, correct me if I'm wrong on this, but what's kind of going through my mind is if we can understand the semantic values of what's being represented by these patterns, then we would be able to be, observing the communication happening and knowing exactly what's happening, from not just from seeing the end outcome, but also knowing what's going to happen before we see it, because we don't interpret what the signal was. Does that make sense?
It does, and I think maybe down the line we could understand at a greater level. It's really, it seems a way off. So one of the things which we do a lot of is we take the Talin and we just alter one of the switches and then look what happens to a cell, how it changes its overall properties. And it's pretty well established now by us and other groups that these switches can move different proteins around inside the cell. So if we can understand the signal which makes the switch go, then we can see the signals moving around, and we can see like the change in the gene expression. So you can start to build this up into a set. the complication is that all of the connections and all of the different signals and all of the interdependencies, because that's the main thing, is all of these linkages and all these like feedback loops of all different layers, like really tight ones and then big ones, and then across the whole cell, and then across the whole series of cells, and they all different levels of complexity. So that would be what we'd want to try and get to the bottom of.
Gotcha. Yeah, that's.
But
Yeah,
one of the main things we want to do at the minute is just with a single memory molecule, understand the rules of mechanical computation on a single molecule, because that's still quite. We've made big advances on that, and we can manipulate them. But how much information can we physically store, and what is the interdependency of those? So we're trying to make different coloured probes which recognise the different switches in the different states, so we can see the colours changing on and off to try and infer what the shape of that molecule is. Because then we want to use those same things inside the cell to see what shape the switches are in. But I'm not looking at the organism level. I want to understand the fundamental rules of this.
Oh no, absolutely. Obviously, that's I think for the audience, obviously we've been talking about some bigger picture and implication stuff, but I think for the audience too, and you've said this many times, like, hey, we're at the very baby steps of all of this, and just starting to, it's taken 15 years of your research to get to the point where it's like, oh wow, here is the macro level implication from a fundamental perspective. It's something I'd love to touch on now. I kind of stayed away from this type of conversation so far, as we're getting into more of the talking about the technical side. But this is something I recognized in the paper, and I would love to. I'm going to share this with the audience, and hopefully you can expand. We've been talking around this. So you said. Quote, the appearance of Talin at the dawn of multicellularity allowed cells to store information persistently by writing to each Talin molecule like a computer writes to a disk, as well as serving as memory. These switches also coordinate cell signaling and provide a way to control the reading of the genome from the periphery of the cell. Now, that question where I'm going with this is, and you mentioned this earlier about the conservation of this, if everything we're talking about is true, does this imply that the function of the Talin protein would have to be accounted for very early from an evolutionary timeline perspective?
Yes, I think it's the level of information that can be written as like these molecules, like the cell molecules and the cell matrix molecules came together about the same time to link cells to each other and to this extracellular matrix. And both of those enable the cell to write information and to talk to each other. Those two cells can then exchange information, and they could synchronize to the point that you could imagine maybe this one could read something and this one could pass that information on. So the development of higher order things. And at the dawn of time, these were like slime molds and things like that. They've got this machinery, they've got this mechanical computation systems, but simplified versions, and they can transition between a single cell and a multi-cell. So you can just start to glimpse when this starts to occur. And what's really nice is that between slime mold and sponges, there was only a few changes to Talin, but the changes were the introduction of regulatory elements. So prior to that, then they weren't as tightly regulated. They could suddenly control this, and then they could start to complexity could occur from these molecules. And one of the things I got asked, I think it was when I was chatting to someone the other week, was about this protein 2,500 amino acids long. So it's a really long thing, but it's really repetitive because these switches, they look very similar. They've got very, very different functions, but in terms of domains, it looks like it's just repeated, and then immediately they've all specialized. So that's conjecture and speculation, but what's really striking is that we've just Talin and its main binding, one of its main proteins which interacts is vinculin. Those two proteins can assemble into like, I don't know, I worked it out once, it was over 10,000 different combinations. So the amount of information you can write into two molecules interacting in unique ways is vast. So you suddenly appear these two molecules, and then all of a sudden you can start to massively exponentially increase the amount of information which is capturable.
And what was the? Can you repeat the name of that protein you're referring to? And like, is that a small one, a big one? Is it as complicated as the Talin? What's can you give a little description of that?
Vinculin. So when these switches in Talin open up, a lot of the switches, like 9 of them, have got at least one interaction site for another protein called vinculin. And vinculin is another linker protein between, it links Talin to the cytoskeleton. So these linkages grow and they mature as you pull on them. So the switches of Talin coordinate the linkages. So these two guys, but because the Talin's got, I think so I don't know why I know the number. I don't know. I'm guessing Talin's got 11 of these sites for vinculin. So as you pull on it, you can recruit up to 11 vinculins. They can cross-link to other Talins. Talins are so you could build like big meshworks just by triggering a process. And then this. So we were even just trying to understand those two as a sort of discrete memory module, which might have just appeared, and to learn the information which that can encode.
That's incredible. The something as you've been describing this, something's really been hitting me, and I think is very often underappreciated by the average person, is the importance of the three-dimensional structure of proteins and how they interact. And hey, this scaffold, even for just scaffold proteins, right? They're just shape, but if it doesn't have, there's 17 different proteins that all bind in a different offshoot, a little side chain of this one. Is that same type of importance of the three-dimensional structure in play with what we're talking about today? I'm assuming it is, but is that something you guys run into as well?
About this three-dimensional shape changes as a function of the force. So these molecules change their shape. These domains open and close. So you've got a protein which does one set of things at rest, and then as you start to move it, it starts to change its shape and switching on and off different binding sites appear and get destroyed. So you're constantly changing the signaling, and that's the mechanosensitive signaling hub idea. But one of the things which we published earlier this year, which I think is hinting, and it's like what you were talking about with the logic gate, is that we've identified during the cell cycle, there's a series of enzymes which make sure it happens at the right place and time. And one of those enzymes, called cyclin-dependent kinase 1, it interacts with these adhesions, but it phosphorylates Talin and changes the order of the switches. So the same amount of force will result in different patterns. So the cell can be constantly modifying the Talin scaffold and changing what string you would generate under the same conditions. So it's maybe I can share this.
That's my. Are you sharing your screen?
I'll try. I failed miserably last time, but
if you're sharing something from your, there we go. I'm seeing it.
So this is just about to come out. This is where we're trying to summarize what the theory is in cartoon form. But what I wanted to show was, so this is Talin in the middle, and then I'm trying to. This is we don't know all the ligands yet. We're trying to work this out. But this is the different domains, R1, R2, and then when they switch, these are the different signals which are turned on and off. The reason for showing this is that underneath each of these domain names, these are amino acids which are modified via different enzymes and via different signaling pathways. So it's not hard to imagine that these are switching on and off changing this pattern, changing which binding sites are available, and that there's a complexity to this. And this leads to this notion at the bottom, I don't know if you still see this, that you could imagine that a certain amount of tension, depending if those enzymes are switched on or off themselves, will generate a different output. But it can all be written into these mechanical switches and how they all respond to force, crossed with like the different cellular behavior.
Interesting. So what you're, I'm not stopping sharing your screen. I'm going to pull up something. Is what you just showed there, is that similar to this?
Yes. So this paper here, this is going to come out in November. This one's so the MESHcode theory is the culmination of all that what we work on in terms of Talin and mechanical signaling, taken the implications of what that leads to in terms of information processing and an idea of memory. This paper here is based much more on the actual experimental data, what's going on, how these switches can flick and alter what's going on. So this is supposed to be, this at a glance series, I love it. It's like really much more straightforward to a poster like I don't know what it is, A2, to explain a difficult concept.
Right. It's kind of it's an infographic.
Yeah, it is, and it comes in an accompanying thing. But this is to try and explain this in easier terms. So I'll stop sharing now because I don't. But just to finish on, which I'll show on this, is that if you do imagine what these structures look like, but reimagine them as strings of zeros and ones, then this signaling which forms at that site, when you switch on the motors and you pull on this and you change the patterns of ones and O's, then that ones and zeros, sorry, that changes the signaling at that site. It changes these linkages, so the cell changes its whole architecture. But it's all fundamentally, it's dictated by the shape of those molecules at the core, which is why we say that these switches are both memory but also reprogramming, why they're changing the signaling. And they can then carry this information forward in time, because if you were to then have that memory module like that with that pattern of zeros and ones, if you stimulate the motors again, you'll pull on that and you'll change this pattern of zeros and ones, and then that will be dependent on what state that's already in. So you can constantly be updating in a very logical, quantized way via these switches. So that's the idea of why these adhesions themselves are the memory modules. I'll stop sharing now.
No, that's very interesting. So you've got, so what you're kind of saying about your paper that's coming out, you said November?
Yeah, that I just showed you there was the finished proofs, so it's going to be out imminently.
Okay, gotcha. So if I understand you correctly, the paper that we've been discussing today was you were explaining the concept, and this one coming out is kind of the proof, experimental proof. So you kind of allude to in the MESHcode theory paper, and this is kind of like, okay, now here is the what we've been doing from a experimental level to kind of back up the theory. Is that?
Yes, and it's also like less hypothesis, more what we actually know, and this is more experimental, and it's basically it's a follow-on, but more of an explainer to put it into more what's known, because the idea of the memory storage in the brain, we know that it happens in the individual cells, we know it happens on the individual molecules, but there's a gap in our knowledge between that the brain could order its molecules in such a way that you could write information into the shapes of them. So that bit is what the MESHcode paper we're talking about today is aimed to propose.
Well, that's interesting. In line with that, some of what I would say is this is close to the end of your paper. You said the complex mechanical coding amounts to a machine code that's constantly running in all animals. From an initial state at birth, the life experiences and environmental conditions of the animal would be written into the code, creating a constantly updating mathematical representation of the animal's unique life. It is possible that consciousness is simply an emergent property arising from the interconnectedness of electrical signals connecting all these mesh codes, forming a complete mathematical representation of the world that gives rise to precise electrical signals that coordinate an entire biochemical organism in the context of its world. That really struck me. Now, I would personally disagree with you on consciousness just being emergent in the context of all this, but I get I totally understand the implications of what you're saying here in terms of at a minimum, it could be representing the experiences of that consciousness being stored and rendered, if you will, in a real-time fashion. Is that fair?
I'd say that the consciousness is incredibly speculative. My wife wanted me to take that bit out because she said there's no real basis for that. But just as raising the idea, but I think in terms of the rest of that paragraph, I quite like it, because you'd develop in the womb, your brain would get to a sort of default state, which we all get to by and large. There'd be variations between person, but generally the circuitry gets wired up and it happens to be the right thing. And then even when you're in utero, but certainly from then on when you're born, depending on where you're born, what happened, environment you tend to be born into, this would then update these patterns of the shapes of the molecules depending on the inputs, what you see, what you taste, what happens to you, what you learn, and connects them all to your output. So I like the idea, and I think it's right that there's a mathematical representation of each animal's unique life. So you've got a default state, and then it optimizes itself as depending on what happens. And where the ghost in the machine is, whether that is piped in from elsewhere or it's emergent, I wouldn't like to guess. But one of the things I would say is that the notion, if this is right, which is what I'm obviously on here to tell you that I think it is and what we're trying to prove, is that we're looking at an ordering of molecules and even to the shape of each individual molecule, which is way, way more complex and more ordered than what we can currently imagine. We're sort of imagining these cytoskeleton forming or whatever. But here, each molecule would be becoming synchronized and written, updated with all this information. And with that amount of order built into these simple building blocks, then who knows what is at that level of complexity. So it may, I can imagine it.
Yeah, the possibilities are rather fascinating. And the point you made about, hey, I don't know if it's being emergent or being piped in or whatever. Either way, I think what we're talking about now is what would be necessary for consciousness. Like if consciousness is going to be in play, then you have to have it at a minimum X is something that could be being determined by all of this. And I personally find this stuff just extraordinarily fascinating, whether you want to go from a idealistic, a dualist, a materialist perspective, all those different things. It's like, hey, we can all have our differences of opinion, but I think at a minimum the macro level implications of all this are quite extraordinary. And one of my personal frustrations, and I've been trying to be nice, not to you in just in the context of the way we've been framing questions about machine code and computation, all these types of things, but I find it very fascinating how many people want to argue that there is no such thing as like actual information processing happening in a biological system. I'm just like, what world are you living in? Like, this is people like yourself and other research. All these folks are, doing this high level research and recognizing, hey, this is what we have to determine. And there's whether or not you want to argue over the origins of it or not. There's no avoiding the reality that this is what's going on. Is that a fair point?
I guess so. Yeah. But in terms of like, again, just going back to the one before, the emergence or not emergence, as a thought experiment, if you imagine a human brain has got the machinery, this mechanical computation, then all the way like in slime molds, they've got the same machinery. Obviously, it's a much more simplified system. And in worms, in mice, monkeys, all the animals, like they've all got this same machinery. And unless there's a specific point at which consciousness emerges, then maybe consciousness is in all of these type of linkages, and it's just the complexity of the calculation or the computational power of how the brain gets more developed and the interdependency of these molecules. So that would be possibly the argument for why it's not piped in, or that it's like, because I think maybe all animals, maybe all life forms are conscious, but it's a pure, you hit well into the philosophy there. But in terms of there has to be some way to read and write information in the brain, whether it's or in life forms, because you've got to be able to learn, like anything which involves learning, you've got to have some form of modification which takes that forward in time.
How could you possibly learn X function if you're not storing the underlying source code that's necessary for X function to be executed? That makes total sense. Something before I forget this, the point you were making a minute ago about changes, after birth and such, seems to me with what we're talking about today raises the implications of, well, nurture obviously, but just the epigenetic variables that are in play in a variety of fashion. I read, I'm trying to blank on the author's name. The book was The Deeper Genome, and John, I forget the guy's name. Anyway, one of the whole section of the book he was talking about were some of the known either negatives or positives in relation to epigenetic variables derived from life experience, and how it was dynamically changing capacity or creating all these different changes. And it seems to me that what we're talking about today could be the underlying this is something in this manner is what's being changed. Now, what the cause was, we might not figure that out yet, but this is what's being changed, which is now resulting in X outcome from a epigenetic perspective.
Yes, I would say to my pet hypothesis, which again is my theory, like what I think is not necessarily, I'm saying it's right, but you'd have these switches which change the cytoskeleton, which would then push and pull on the nucleus to change which genes are exposed. And then the epigenetics would be a layer on that where there would be different markages. So you would write information, the cytoskeleton would cause alterations, and then that would be the epigenetics would be a way of like helping to stabilize those patterns. So mechanotransduction and the alteration of epigenetics as a function of mechanics is really well mapped out in single cells. The problem there, again, is this lack of the higher level order, that it looks kind of a bit random because it depends where the cell's pushing and pulling. Whereas in an ordered system, you'd have this perfect pipeline from a mechanical and a chemical input coming together, altering this cytoskeleton, altering the nucleus and the DNA, and then the epigenetics would be that. So this whole epigenetic code, genomic code, mechanical, they'd all be part of the same overall.
Okay. That makes sense. One of the analogies I've thought about many times, and not just in relation to like MESHcode versus genetic code and such, but other codes. It seems to me like we got to figure out where the API, the equivalent of the API connections are, in terms of all these different systems that are working together. And they're either the codec might be slightly different or might be different, but the end outcome is it's being translated into a uniform one that's being understood by all the mechanisms in the system. And just to me, otherwise, none of this makes any sense. If that's not happening at some level, then there's no way that these digitally encoded commands could possibly be executed. There's like in my opinion, there's no logical way you could do that unless you have and you kind of alluded to in the paper, the adapters and the decoders that are necessary to recompile whatever the message is to result in the end function.
Of course, yes. And that's where again, my pet theory on this is the mechanical computation, that the cell makes all of these motors and all of these polymerizers, and then the interdependency of those, how they all work to balance out and to minimize all the time, is an actual computer with a level of complexity and much greater than what we are predicting at the minute, because we don't consider how ordered and how meticulous that might be. But that it might be that this always reaches a state, and the cell's got a default machinery which exacts the program, and then depending on these patterns it runs it slightly differently. And that's where you could imagine that from a single cell you could build and build and build and get complexity. The challenge is how you get to a single cell, in my opinion. But the cell's got this machinery and this computational power way above what anyone would predict at the minute. And I think it's this cytoskeletal machinery, this mechanical computation.
So with this, if what we're talking about now is the underlying necessities that we're talking about right now and the importance of it, does this put the relevance of the membrane formation, the structures, and all those kind of things, does that take all that to a new level of both importance as well as complexity in terms of the interactions and everything of the cytoskeleton?
I guess so, but the whole thing is like has been optimized over many years. Even if wherever it came from, it's still finely tuned against its physical environment and optimized, and every single part of that system is heavily optimized. And there's a huge, like, irrespective of where anything came from, there's a huge selective pressure on things all the time to be as efficient as they can. So yes, I think that's why all of these things now look so well. And then you make a single point mutant in an ion channel or an ion transporter, and the system will break because everything's so optimized, and every bit of it's got different levels of interdependency that it's so tuned. So.
Right. Yeah. One of the systems I find very fascinating is the potassium ion channels and sodium ion, and the little packet, like there's a little storage unit over here to make sure you got enough ions for X, and all these different, and the equilibrium state balance, all that stuff just I find extraordinarily fascinating. But in line with that, the voltage gate meters of those channels I find to be
Yes.
just kind of mind-bending technology, if you will, from a nanoscale. And the one before we and audience, just for a second, we're going to be wrapping up here in just a few minutes in terms of the back and forth we're having right now. We'll start getting some of your questions. So if you guys have questions you want me to ask the good doctor, we got in for about maybe about 30 minutes. So we'll see if we can get some of your questions in and take advantage of him while he's here. The one of the things I want to talk to you about to kind of wrap up is we've been using the words mechanical signaling, all these different things, right? But very often folks make the argument of stuff like, oh, and I'm drawing a blank on the guy's name. He's a philosopher of Richardson, I think his name is, philosopher of biology. And he wrote this paper about how the cell is not a machine. And one of the arguments he makes is that, because things change shape, therefore we can't call them a machine.
Yeah.
And so ultimately the reason I'm asking this question is, and to clarify a little bit more, is to me, it seems that just because on the molecular level and the nanotech that we're observing and that humans are also creating can change shape and be dynamic, that doesn't remove the premise of a mechanical function. Would you agree with that? And if so, can you describe a little bit of, as we've been doing, but like the general concept of people in your arena in terms of their perception of what mechanical means on a molecular level?
So I think that debate of whether it's a machine or not rages for a long time. My opinion is it's got machine-like properties because it happens so reproducibly. And the big bit missing in all those discussions with the philosophers and stuff is that and what I'm trying to bring new to this is that this level of order in the system suddenly makes it much more machine-like, because you have to have this order for this machinery to work, is the hypothesis. And one of the cool things about these mechanical switches is you can have lots and lots of stuff coming in and out and lots of things whizzing around the cell. But at the end of the day, that switch is either in the open or the closed state. So you basically buffer against chaos and you buffer against noise. And it means that in all of the stuff going on, then you can either be in that state or that state. So you quantize all of this chaos into discrete steps, which then have switch on different programs, which then have that. So there's this way of generating order in what appears to be a chaotic system. And the shape changing and all of that, again, that's the artifact of looking at a cell in culture. Basically, when you look at a cell in culture, you're looking at a cell in a migratory mode, because it knows it's not where it should be. So it's trying to crawl around, feel where it should be, trying to work out, like normally if a cell was in the wrong place, it will trigger apoptosis. It will say, oh my goodness, this has gone terribly bad. I'm going to kill myself, and it goes, and then it will die. If that's the only way you ever look at a cell, then you're going to think that this is not as ordered as it is. So that would be the idea. So I'd say that I don't want to get bogged down on the philosophy of arguing about whether it's a machine or not, but I think it's got machine-like properties, and I think it's an ordered system which has got switches which encode information.
Interesting.
Does that answer the question? Maybe.
No. I totally follow where you're going. I appreciate that. Now it's very interesting to me that you and other people, lectures I've watched and other PhDs I've had interactions with, many have talked about the differentiation in vitro versus in vivo, and that the fact that we're just now starting to have the technology available to, on some level, be able to observe things in vivo is creating an entirely new dynamic of implications and the things that you're talking about, right? Like I watched a video that was created a couple years ago where it was like the first time they'd been able to observe a motor protein in real time. And they'd been able to take pictures and like create a video of it walking across a microtubule, right? Obviously, there's been animations, computer animations of that for years of that process, but now they'd finally been able to actually visualize it for real. And those kinds of implications, like I said, I think this stuff is extraordinarily fascinating. I can only imagine the cool things you get to see on a daily basis.
Yeah, I think that's it. Like to make a life, you need movement, and the movement is caused by things which polymerize. So you can make a filament which grows long and gets small, and you have motors which actually move. You give it energy, and it actually generates motion. And if you connect all those movements up together, then you can synchronize a whole big system, which is a cell. And then if that cell can synchronize that with its other, then they can move as a 2-cell. And from these very simple rules of motors, polymerizers and switches and things to coordinate all of that's life, in my opinion. So.
That's amazing. Well, Dr. Goult, I greatly appreciate your time and undergoing interrogation with me. So thank you very much for all that. So for the audience now, I know some of you have been sending in questions, and we will get to those here in just a second. But if you want to ask Dr. Goult any questions, make sure you tag me very, very quickly, because we're not going to keep him for too much longer, because it's weekend time for him. He's 6 hours ahead of us, and we'll make sure he's able to get off and have a good weekend, get ready for his trip. And we want to make sure he also doesn't have to explain to his wife what he was goofing off on YouTube. So, all right. So before we get into the questions, is there any like final points you'd like to make? Anything you think we haven't covered, or you'd like to kind of leave as a final touch with the audience?
No, I don't think so. I think it was an interesting discussion. I think there's a lot to work out, and yeah, it's a great job to be in.
Okay, so let's see. First question: is this extracellular matrix controlled by genes, and what role does it have or not have in embryonic development? This is from Andrew Cumming, I believe.
Yeah, brilliant question. So yeah, it has an essential role in development, and the cells make the ECM. And I don't know if you've ever seen a dog making its bed, where it pushes and pulls and tugs on it. Oh, yeah. And they do that. So if you look at a cell on some ECM, it pushes and pulls, and then there's things called fibroblasts which go through the matrix and push and pull, and they balance it out. So yeah, basically, how I picture that is they're writing information. They're getting that matrix into a specific pattern of information which the cell can then interpret, and it's absolutely essential. Yeah, anything which screws up matrix formation, or any of the integrins which grab onto it, then they have massive embryonic, nearly all of them are lethal, so embryonic lethal. So.
Interesting. So there's definitely this stuff better be working, or we might have a problem. And even
and the matrix, there's a whole institutes working just on this matrix. It's like, it looks like a cargo net, but there's lots and lots of proteins and lots and lots of signals in it. And you're probably familiar with collagen, because that's one of the main matrix proteins, but there's things like fibronectin, and they all make this different meshwork with different patterns. So I think it's patterns. It makes different patterns in that. And the cell, you could imagine that the cell could alter that to alter its coding inside. So yes, I think it's really, really important.
Interesting. Okay, so up next we've got a Utopia Buster says, is there any relationship with Penrose's OR theory?
No. Well, no. So the good thing about the MESHcode theory, in my opinion, is that it's based on biochemical and biophysical rules of how a protein will behave in response to mechanical force. So all of the bits of it are based in the reality, and it can be coordinated via simple rules of turning motors on and off, which exist and has been shown to exist. The complication with the ORCH and the quantum in the microtubules is, how do you have wet quantum where, and how do you write that? The microtubules are changing, they're doing the state, and how much information could you encode in that? Is that a single binary thing? Here we've got many switches, so the different patterns and combinations of those switches. So no, I don't think so. It's the short answer. I've heard them banded together, but like MESHcode and quantum microtubules, but I don't think so. And I think the good thing here is that we should be able to prove this. But I can't imagine a route to prove the quantum stuff. Not to say that, that might be a limit of my imagination, but
no, it makes sense. I think, correct me if I'm wrong on this, isn't it aren't they saying it's like just the asynchronous microtubules that we do in the quantum function? I think I forget. There's some delineation of like only this type of microtubule could potentially be doing the computation. But I agree with you. It's definitely something that's difficult to analyze, for sure.
Yes.
And
so there was a really interesting comment from Crick a few years ago, and he was saying about it would be hard to imagine memory molecules because molecules don't last for many years. Like they don't last for the duration of how long you need information. But the cool thing about the MESHcode theory is that these are adhesions, they're complex, where there's hundreds of molecules which are stabilizing those patterns and reinforcing them via like these positive feedback loops. So these patterns will be able to tolerate, if one protein was to come away or one switch was to go into the wrong shape, it would refit because it wouldn't be consistent with the whole thing. So yeah, I think that's why I'd be. I think these provide a meaningful way to encode information.
Interesting. Okay, so this is super chat from Doki Doki. Ben, this is an inside joke. He says, "Would you describe DNA like a glass of Diet Coke?" Thank you. The reference to this is there's a gentleman who I debated last year who argues that the code in DNA is no different than the differences between Diet Coke and regular Coke chemically, and it's turned into an ongoing.
Right.
It's turned into an ongoing joke of like, what are you talking about, man? Like, don't be comparing the complexities and the importance of DNA with, oh, it's just like Diet Coke, All right, Jamie Russell.
I don't know about that one, but
yeah, well,
everything's like Diet Coke from a certain perspective, I suppose. Like,
basically, to put it in context, this particular individual was adamantly arguing that there is no such thing as code in DNA, and like it doesn't exist as the implication. But anyway, Jamie Russell, thank you for your super chat. He says that was about consciousness. It is emergent by design. Could you speculate about the implications on future tech, i.e., AI, medical advancements, etc.?
Yeah. So, short answer, don't know. But if this brain is this massive, complex computational device, as everyone seems to agree it is, then if it connects all of the inputs, everything you smell, everything you touch, everything you do, and all your other thoughts with what you've done in the past and links it to outputs, then maybe that whirring is this computation. If brains do wire up with a very uniform wiring structure, wiring pattern, which is written into the coding of the cells, then maybe you could imagine that it'd be possible to extract information from it. In terms of consciousness, no, I don't know. Because we don't know where it is or what it is, and it's hard to find even correlates of where it is. But if it is just the processing bit of the input-output machinery, which is, yeah, I don't know. That doesn't really answer the question, but yeah, I don't know.
well, I think one of the other points he was probably asking about too was, and I think you actually alluded to this in your paper in relation to like medical stuff. There's a lot of implications in this in relation to like cancers, right? If of cancer treatment or causes?
Yeah, because I think in terms of like this mechanical signaling goes wrong in cancer a lot, because one of the things which can make a cancer particularly aggressive is if it starts to deposit a lot of matrix. It gets really, really stiff. Then the cells detect that and they think this is a time to grow and a time to start crawling. The signals get corrupted and it becomes migratory, and that's downturn. So this balance between what it wants to be, like, it's kept in check by its physical parameters. If they get changed, then yeah, in cancer that's really bad. So if you get fibrosis of your tumor, then like in things like pancreatic cancer, that can be particularly nasty because the cells become so migratory on it. They get the wrong set.
And pancreatic cancer has like an extremely high death rate, doesn't it? Like, I mean, it's just the survival rate's basically nil almost, isn't it?
You don't want to get that if you can help it, yeah, because it's really, really, it's brutally aggressive, and their cells are they get reprogrammed, and then they go off and they spread so effectively, and it's a horrible. So we're actually trying to work on that to try and work out. It's definitely to do with the stiffness of the tumor. If you make it suddenly so it thinks it's on a really soft substrate, it can be just as bad. It's like you want the stiffness to be normalized, so it's a nice to keep them in check. And then once they're kept in check, then you can treat them with more classical cancer drugs. So that's what we're trying to see. Is there a way to do sort of combinations to limit to stop them getting so reprogrammed, and then also to treat them? So yeah, that's
gotcha. So you're trying to figure out the ways to plateau the reproduction and the metastasis, and then be able to annihilate them, theoretically annihilate them with more traditional treatments.
Yeah,
makes sense.
I would keep them where they're in a nice bit where you can remove it before they start going walkabouts. Yeah.
Okay, like or even just like straight up cut it out. Gotcha. That makes sense.
But by the time
The
they've spread, you're done for, because they go off and then they make their own niche elsewhere and start to grow uncontrollably there, because they reprogram as a function of this.
Gotcha. All right. God is now here. Thank you so much for your very generous super chat. He says, looks like one Evo atheist visited the live stream to give a thumbs down to science. That is one dependable, testable prediction. I got a good chuckle out of that one.
Did I get a thumbs down? Oh dear.
Well, you got one, it looks like.
Oh.
There's a few folks. You got 21 thumbs up and 1 thumbs down. That number's going to go up, trust me. This is early. Usually I do streams much later in the day, so there'll be a lot of people that come back and watch this later. And
Okay.
trust me, they will appreciate our conversation. There's 4 or 5 guys that I know right now that they stayed up way too late last night doing some research on another topic, and I know they'll watch this later because I got a message this morning of, oh yeah, your interview's at 10, isn't it? 10 a.m. our time. I'm like, yep, where are you at? I'm going back to bed. I'll watch it later. Okay, so let's see.
One of the things which is cool in the paper, we didn't touch on it today, was that it suggests a theory for why you need to sleep and why those guys won't be functioning too well today by doing their all-nighter. Is that this notion that maybe if this is physical data storage in the brain, that maybe the reason for sleep is that you physically write the information into your cortex and update it. And that the REM sleep with the electrical activity might be the motors being switched on and on to update these patterns, so that the flow of around the brain is updated with this information. So that was another, yeah.
That's interesting. A couple years ago, I read a study on the cognitive function, like optimal state in relation to sleep, and I forget the exact numbers, but it was like less than, I think, 5 hours and more than like 9, you had equivalent, pretty close deprecation in cognitive function, like executive functions, all that kind of stuff. And then it was like 7 hours and something, I forget the exact numbers, was like the optimal range. One of the things I found very fascinating, though, was the less than 5, you had the equivalent, not just in your brain, but like just overall motor response and everything, was the equivalent. I forget the exact number. It was like having a blood alcohol level of like 0.0 whatever it was. You basically you had the same capacity as if you were drunk, basically, with the sleep deprivation, or you were in some kind of same too much as well.
Yeah, because a lot of it, like when you have your day-to-day stuff, it all goes into your hippocampus. It's computed and thought about and processed. But then it has to be, that's like you could imagine it as sort of like RAM, and then it gets written into longer term storage. And it also gets consolidated with all your existing memory. So there's this notion I propose in the thing, that the reason when you first go to sleep, you're going to really deep sleep, and that enables everything to finish from the day, and like all the calculations and all of the input and everything to reach a sort of steady state. And then you have this higher energy REM sleep where there's a lot of electrical activity, and that's where that information is then written and processed and written into your brain. And then you go back to sleep really deep, you go into a deep sleep again to let that finish off, and then you have multiple cycles of that. And it's basically like database management is what I try and postulate, because you've got to rebuild that data to make it consistent with what you already know, because that's one of the amazing things about how we function, is how we make things consistent with our own world and our own understanding of the world.
So it's almost like we've got a you're cleaning your cache, if you will, then keep storing all the important stuff, deleting stuff that's not, making sure everything is put into the appropriate pathways and addresses and such, in this context, in relation to like the flash memory. But do you think that what we're talking about right this second has implications in relation to, from an educational perspective, of why getting a good night's sleep, especially as like for kids, right? Because they got to get up in the morning, go to school, and all those kind of things, that if they're not being able to get enough rest and that deep sleep REM balance, that it might. This is hypothetical here, but do you think that might impact how well they learn something, or how fast they're able to learn a concept?
Yeah, I think so, because you've stuck your RAM's full at the start of the day, where it's not fully cleared and you've not fully consolidated what you've already got, so you're already running with it. Like if you start from a fresh start and you can start to build all that stuff, it basically it almost like not quite the same, but like almost gets like fragmented because you've got the information from that day which you've not built in. So it's after a while that becomes hard to read and to perform with, and you notice it. I mean, when you do an all-night or you go out for a heavy night, then you're not as cognitive the next day. You're not as useful in terms of.
No, I totally understand. Yeah, it's definitely a problem, which I think I would wager that, especially when you were working on your PhD, you probably experienced a little bit of sleep deprivation on occasion with all your research. Okay, Tony says, what's his opinion on the chirality problem and selecting the 20 correct amino acids for life through purely or in prebiotic environments? That's kind of obviously an abiogenesis question. I don't know what your thoughts are on that.
So I don't have a strong thought on it either way. There's obviously, however you think about where everything came from, there's a huge gap in terms of the understanding of where these things assembled and how you got to it. And from a cell onwards, I can imagine how things could work. It's how you get to that cell with all of its working parts and interdependencies where I struggle. So I've got no strong feeling. If I had to say what my preference would be, it'd be almost like, as I was saying the other day, I was talking to a Tangelo, where if we can fly to Mars now, if we were to seed that with some cells and wait 10 million years, then there might be all sorts of different life structures there. Like, because it's how you get to that cell in the first place. And maybe a cell was developed, a cell emerged somewhere else and came to this. Because the bit I struggle with is, and I'm not saying it's wrong, I'm just saying there's a bit missing in the understanding of how I can picture it, is that lightning hitting a sulfurous pond, how do you capture and how do you improve on it? It's not like that's wrong. It's like there's a big gap between that to that. And so there's nothing to fill that gap. Everyone fills it with their own working hypothesis. Right. I don't know, but I think from a cell onwards, I could imagine evolution from a cell.
Gotcha.
That doesn't answer the question. I'm shaking it because I've got no idea and I've not got a strong opinion either way.
Well, no. Actually, I appreciate the fact that you say you don't have an opinion either way and having a hard time, figuring out a viable, position on it, because I think a lot of folks take a much more aggressive position. Oh, we've totally solved this, and I'm like, there's a ridiculous amount of steps we have not accounted for in any way, shape, form, or fashion. That's seems to be a very major leap. So Dr. James Carter just joined us. He's also a PhD biochemist. He just popped this up here. Says, does Dr. Goult think that the effects of ethanol in the brain might have to do with the interface of hydrogen bonding and Talin interactions?
Yeah, I thought this crossed my mind, because one of the things we first sought to do when we were doing the single molecule, pushing and pulling on it, was to try and do that and add some ethanol in. And we might still do that experiment, actually, but then I got more moving towards that maybe it's affecting the quality of the signaling and actually disrupting the receptors on the outside. But yeah, ethanol clearly makes it work less well, or better well in some respects, depending on how you feel. But it's a good question, and I don't know the answer for sure, but it would interfere with the hydrogen bonding for sure. Yeah.
Interesting. Imagine you two.
But why
have a good combo about this?
Why be specific to not screwing up everything? Maybe just in the bits which are currently doing a lot of the activity and a lot of the changing. Maybe the ethanol tilts that slightly, makes it less effective. But the stuff which is changing more slowly, maybe that's not as affected by. So yeah, it's actually a good shout. So.
Interesting. Now, this is an interesting question from Andrew. I think in your conversation with Tangelo, you guys had talked about how there weren't really any precursors to Talin, but he says, you mentioned how the Talin protein is made of many repeated domains / segments. Is it possible that duplication and divergence could explain this?
Yeah, so the interesting thing with the switches, most of the switches have got this unique fold, which is only in Talin. So this fold, like, it's a 4 helix bundle with an extra helix which comes on the side, and that extra helix is really nice because it makes it so that the ends of the molecule, the ends of the domain, I can't get my hands right, it's really weird with the mirror, either end. So you've got your domain, and then on either end you've got the end. A 4 helix, both ends are on the same end, So the addition of that 5th helix really changes the properties. So, but then every single one of those domains has got the same structure with a couple of exceptions. Then the main exception being the very, very last domain, which is actin binding, but that's in all animals. That's in yeast. That's a very common fold. But then the 12 switches after that, or the 10 of those switches, have got this unique fold. So maybe it appeared and then it duplicated fast if it was to be. But then once they've got the same fold, but they've all got different jobs, so they interact with different stuff. So even though it's got the same fold, so yes, it could definitely have come from duplication. But the interesting thing is that it's every single talin in every single animal cell from molds upwards. They've all got the same 13 switches. The number, and they're all in the same order. You can tell from looking at a slime mold which switch is which. So this pattern has been perpetuated all the way through all animals. Okay, so if it is a data string, a data molecule, then the order of the switches would be presumably important, and that order has been highly contained, and it's never changed. It's never had 10 switches and then another 15. It's always been these same 13, exactly the same, which is quite unique in terms of molecules, in terms of how aggressive the conservation's been.
Right. So it seems to be kind of what you're saying is, yes, theoretically, I guess it could have come from duplication. However, that would have had to have occurred prior to slime mold, as an example, being in place, because everything from that level up has the 13 switches. There's not this one has 7, this one has 9, this one has 11. They all have 13. So either you have to say that it evolved prior to all of this happening, and then it just stopped at 13, or it was 13 from the beginning. Is that kind of the...
Well,
again, I'm not saying I'm saying it's 100%. I'm just saying like that's a
pre-slime mold. There's no records, so you could make up anything in that. What happened? Here be dragons, I think is what my PhD supervisor made when I had a 2-point graph. It's like, who knows? But I mean, it'd be easy to imagine that you could build this up. So you could say it just appeared, or you could say this domain, there's a lot of duplication, because there's loads and loads of helices in the cell, and maybe I don't know. So you could argue that both ways, and I wouldn't want to be drawn on which way. But the fact is, of all the records which we've got access to, maybe we'll dig a deep hole in the Antarctic at some point and find stuff and say, oh, it's only got 6. But in all the stuff where we've got access to its DNA, so we can tell what sequence the talin is, it's always got the same 13.
Gotcha. No, that makes a lot of sense. It's kind of a, yes, I can imagine, as you said, here to be dragons, but it would be an assumption or an assertion, not something that's empirically proven from and it'd be quite surprising. The data you currently have access to anyway.
Made a protein like that, or maybe I don't know. It's like
no, that's very interesting. Okay, so Utopia Buster says, would you consider consciousness a fundamental aspect of universe nature? Can memories as information actually be lost?
So the first question, one possible thing, and I'm not a big advocate of it, but is this notion of panpsychism type thing where everything's got matter, consciousness at some level, but the ordering of this, and maybe because I'm proposing every single molecule in a head is either in a specific conformation or is moving between 2 different positions. So they're all ordered in a really high way, in which case then maybe that leads to a higher concentration of this. But I don't know if that's actually true, but you could imagine it. In terms of memories being lost, absolutely, because if you scramble these patterns so that they no longer make sense, then that information would be gone. The same with the scratching the CD. The CD, you've lost that information because the ones and O's don't exist anymore. So when you're playing your favorite track, you hit that bit and the words are gone because it's no longer there to make sense, and you can't retrieve that very well unless you get another copy of it. But so I think memories can be lost, and they can be overwritten as well. And I would also say that forgetting is a really important part of memory, because one of the things that you're doing all the time is constantly updating it and building it up with the new information and the new connections you've made. So
makes sense. Okay, Andrew Kaufman, great member. Thank you so much, Andrew, for your support of the channel as well. You
I should
say,
get a cut of that.
what does Dr. Galt think about head cheese or smart cells? Can they be programmed eventually using an understanding of mesh code?
I don't know. I've never come across those two terms. So one of the things which we're trying to make at the minute is actually build material out of Talin using these switches to actually build a meshwork like a gel. So would that be a smart gel? But then you'd need the beauty of the system is that the motors switch on and they pull a finite amount of time to flick a switch, and then so in principle, yes, but I would imagine you could easily make a gel which has got these switches in to report on a single thing, like has it been stretched, yes or no. But whether you'd write what level of information you could write into a unrestrained gel might be quite limited. But that would be one aspect I could imagine.
Right. Well, I think my personal opinion is we're just barely scratching the surface of what's possible with the programming that is available, or the potential for programming that's available in synthetic biology. I mean, let's be honest, we've barely had the up until CRISPR, what we were being able to do was extraordinarily limited, and best like maybe we'll be able to pull off, when have an idea of what we wanted to accomplish, but actually being able to execute it on a digital level was not all that feasible. And of course, as you know full well, I mean, the programming technologies that are available for DNA synthesis and everything else is nuts on what's being researched. I don't know if you follow this stuff, but some of the guys over at Cambridge, they've written a custom language specifically for DNA programming, and there's another group in India, and I think MIT's also done one. Like one of the groups, I forget which one now, they took the Verilog code that we usually use for chips and created a new version of it specifically to basically, you can write in Verilog code, and then it turns into a DNA sequence, and then you can send that off to be synthesized for a custom function. And I know there's different variations for those kinds of capacities, but that's just kind of, when you think about that kind of opportunity, it's kind of mind-blowing of the potential opportunity as well as the ramifications of what's doable. And I would assume that same premise. I mean, good grief, we could figure out how to program a mesh code. That's a I would wager that's probably a whole nother level beyond even what we're doing with CRISPR at this point. But
but if you look at the difference between a gorilla and us in terms of the DNA, there's a relatively few number of changes which have led to a several orders of magnitude difference in the computational powers of these systems. It probably because of the circuitry of the brain, to be honest, but still just more complexity, more circuitry, more higher order of that. So it's hard to imagine that you couldn't have a similar set of changes which would lead to something which would make us look as simple as my dog, basically, in terms of the level of intelligence, because that's what you see going back through evolutionary time. Like as they get more complex, the changes are quite small. Somehow the change in the coding of the individual cells gives rise to an organism which has got a vastly more complex, like orders and orders of magnitude more capability.
Now, when you say the differences, are you referring to like the protein, like the gene sequences, or are you talking about the regulatory factors as well? And the reason I asked that question is some things I've been reading recently are the discoveries that, oh wow, we thought there was these amazing similarities in the coding regions, but then we're finding out that the regulatory aspects are dramatically different between organisms. Do you have any thoughts on that?
Well, I think that calling it junk DNA is like peak human arrogance, because we don't know what it is, therefore it's junk. Is like the hubris of which we can have as a species is quite vast. But in principle, like the genes themselves, in terms of coding, they might just be the objects or the things you can call in certain orders in response to certain signals. And it would make much more sense that the coding is the other bit, like the regulatory factors or the localization factors, so that you pull different bits to the edge of the nucleus in response to different signals. So I think that junk is where the coding exists, is the short answer to that, I'd say. But I've got no evidence for that, but that would make
You don't happen over on your side of the pond. You don't happen to know who Dr. Nessa Carey is by chance, do you?
No.
I think she's at I think she's at University of Edinburgh, perhaps. But anyway, she wrote a book called Junk DNA.
Oh yeah,
And
I know that book. I've not read it, but is it good?
it was very interesting. She kind of makes that point throughout the entire book of just like, okay, we are doing nothing but discovering new functions. We're just like I forget the exact number. One of the points she was making is that it was a huge percentage of the new diseases that are being discovered are the result of mutations in regulatory regions rather than coding regions or like gene sequence regions. And I thought that was rather interesting. And then something else I came across recently, this goes back to the cognitive aspect. This is a relatively recent paper. I think it was 2017, maybe. Anyway, they had come across one of the points of the study they found was that there was like a 30% variation in expression levels of different aspects in like chimp versus human brains. So like, oh, it was all these very similar sequences and proteins, all these different things, and they're like, oh wow, this one is way more expressed, and this one's downregulated, and all these different things. And it was the regulatory variations that they were finding that were dramatically different versus we're only 2%, 3, depending on who you argue with. It's 12 to somewhere between 2 to 12% variation in chimp and human DNA, and they're like, wait, there's 30% in this portion of it from a regulatory perspective. This was specifically in relation to cognitive function.
Yeah, bit contentious, but I have a bit like an aversion to all of this, where going in blind and just dissecting enough brains will work this out, like, because the actual core bit, the bit you won't see, and there's so many, like we're working this out at the fundamental level and at the cell level, but then hoping we can scale it, and then we can look in an animal and we know what we're looking for. Like all of these people are basically looking for associations between some variables and saying, oh yeah, this has changed, and I think there's a lot of, I should also, because I'm going to get contentious, but there's a lot of blind spots in neuroscience. Like there's a lot of blind spots in a lot of fields where it's assumed that this is known, whereas the actual evidence or the linkages to show that this causes this is a relatively small amount of evidence, which is then being extrapolated really, really far, which is what I'm doing here. Apart from we're trying to work at the individual molecule level and then the cell level, and then a group of cells, a sheet of cells, and then an animal, and then getting the human diseases and coming backwards and trying to build it up across the different levels of explanation. Because if you just do an MRI or you just do a lobotomy or a scalping of a monkey, what you learn, so
you're not getting in the as we were talking about earlier, you're looking at in vivo versus in vitro and then expecting to, oh, this must be what's happening, versus, hey, you're looking at a broken system right now. Why would you expect the
and we don't know what we're looking for, but there's an association here. This protein's changed 30%, therefore this protein must be doing this, and there's this sort of leap in, like, this means this, whereas the bit in the middle is the here be dragons again. Like, it doesn't necessarily, it might be that this is a result of something else, which is also, so I think it's, anyhow, my wife's a vegan, so we try not to do, I don't do any animal stuff really. We can work a lot of it out. We can work a lot of it out by just understanding what's going on and the rules of the game and what the actual, how the building blocks and how the machinery works, and that's our focus.
Well, that's one of the benefits of at a minimum the human level of consciousness, right? We have the ability to imagine and to extrapolate things based on, I mean, what is forensic science, right? Like forensics necessitates that aspect of consciousness and be able to imagine what must have happened and then be able to test the imagination to find out if you're true or false in the end conclusion. Real quick,
it has to be consistent across all the layers of explanation. Any idea, there's not going to just suddenly be this thing here where this electrical signal is what's happening. There's got to be something which happened, and it's got to cause a meaningful biochemical change. There's got to be some way of writing this and changing this system. So anyway,
makes sense. We're getting close to our stopping point. So let's see.
I don't mind saying another 10 minutes of this stuff.
Okay. This is Andrew was talking. He says, Dr. Thomas DeMars, University of Florida, rat neurons grown and taught to pilot a flight simulator. People have called the grown neurons head cheese. Okay. That's what he was talking about earlier on the.
Right.
That's interesting. Amazing what you can do with rat brains, apparently. Appreciate that, Andrew. And then other Andrew says, might wrap on this one. One last question for Dr. Goult. Does he have any general thoughts in the field of evolutionary developmental biology?
Only pretty much what I've said, that I can totally imagine. I don't imagine from a cell building up the complexity with the increasing computational power of parallel processing between different cells with the matrix. So you could imagine that you could build complexity from that state, but up to there, no, I don't have no. Is the short answer. I don't know what happens to get to the cell.
Yeah, Jeremy England wrote an interesting he's written papers in his one of his books on the abiogenesis problem, and one of the things he discusses is the barrier just between like prokaryotes and eukaryotes from a thermodynamic perspective was like 200,000 times greater, I think was the general number he put on it. And obviously there's a whole nother gigantic barrier between, random chemicals and even a simple cell.
But maybe I should clarify that, because in terms of, by saying that, what I mean is in terms of the hypothesis for how that comes with the lightning hitting the ponds, there's not a sufficient way. But there's not to say that there's ways where this complexity could emerge, because one possibility is that with these 4 proteins like talin, vinculin, actin, and maybe just with those 3, you can already start to assemble vast arrays of complex information into the shapes of those. So it could be that you just need to generate a few building blocks, and it might exponentially increase in complexity, and it might be that you can build really fast. But at the minute, I've not seen anything to like how you'd get to that, even to that stage, or how and a lot of the steps to have change over time, a lot of the steps you might have local energy minimums where it's actually detrimental on the way to being better. So there's huge gaps, and that's not to say that I think they're impossible or that there must be a creative step. There's just big bits of the puzzle which are missing completely from any discussion of this. So anything to fill that gap has to be faith-based, because you've either got to assume that it can come from absolutely nothing, or you've got to assume it can come from some sort of creator. But that bit we don't know. And if you think you know that, then it's faith. It's just faith.
Yeah, the point you're kind of alluding to there on the gaps in the knowledge, a lot of times people want to say, oh, well, we'll figure it out. And I'm like, from my perspective, especially the more we're learning from the technological aspects and things we're discussing today, and like you made the point, hey, oh yeah, if I have Talin, if I have these other things that we know can do information processing, then you could see it. And I agree with you. Like when I look at what we're discovering with cells and mechanical computation, all these different things, like, okay, to me, there's a lot of aspects of what we're trying to recreate in artificial intelligence systems that we're seeing happening in biological systems, right? We're seeing very similar types of things, except better than what humans have created so far. And I'm like, okay, well, if that is the case, then theoretically I could see how things could improve and get better over time, all this kind of stuff. But that's assuming you have the initial AI system, right? That's able to be accomplishing all these functions. And then I'm like, okay, what is the necessities for those to exist? And you start, to me anyway, as you keep going down further down to the root necessities, to me, I'm just from my own self, it's not that I don't think that we can potentially figure out a way to do it as humans. To me, just the premise that it can happen without the underlying necessity of the intelligent agency, to me, seems to be here to be dragons, so to speak. For me, even if you want to argue that, hey, after this is done, then more advanced things get developed. Okay, that's fine. Although I think from a probabilities perspective, you get into some crazy unlikelihoods of just what are the probabilistic resources of the universe. Even if you account for a 13.8 billion year old universe, is there enough probability resources to account for like the functional bits? And that's you talked about this earlier in context of like the Talon's 25 in the ballpark of 2,500 amino acids, right? You think about that from a number of functional bits that are necessary for that to exist, then you're so far beyond the total random search probabilistic resources of the universe.
Yeah.
And those are the kind of things to me, when you really contemplate, it's like, this is some kind of mind-bending stuff to think about,
I could totally think that from a cell, like we could plant cells, they could grow into different stuff. Like someone could have put a cell down and then just watch what happens. And in the context of the whole universe and the how many billions of years, the fact that we don't think that if this has happened here, that it couldn't have happened somewhere else a long way before, and then we might be an experiment on someone's desk in their in their world. So it's not so the main thing is like I can imagine us seeding life on other planets, so I can easily imagine something else seeding life on this planet, and maybe there's conditions somewhere else, or conditions which we don't know of here, where it was very, very different, and there was a lot more. Because even if you have something which came, like, even if that just moves the goalpost to another time or another planet, but this sounds like we've had like 6 beers or something and shooting the breeze, but
well, no, I mean, hey, well, no, to me, the point, I mean, heck, let's get real. I mean, Dawkins brought up the panspermia premise himself. I mean, like, it's not like it's been something that other folks haven't thought about in relation to.
No, I'm not saying they haven't. I mean, just that, I mean, you can imagine that. You can't imagine like nothing.
Right. Well, exactly what I was going to say. Like, yeah, so in some contexts, it's kicking the can down the road, but the other side of it, it's well, at the same time, I don't really see the plausibility of it happening without something like that, even if I push it off of the planet, or even if I want to avoid the premise of, God being the initiator. It's for me, and I think, and for you and for many others, it's very difficult to, especially in my opinion, as our knowledge continues to grow and the necessities and the underlying technological components that we're starting to realize are in play. I remember reading Hubert Yockey's paper from the early 90s, I think, and he was talking about this in relation to abiogenesis, and he was talking about how the fundamental principles of information and communication theory must be in place since about the initial stage, and that it was something that must be accounted for in everything that all the research going into abiogenesis, of like, because if we don't account for this, was the point he was making, then we're just jumping 17 layers past where we need the starting point, and being like, oh well, assuming all this happened, then my theory is going to apply from this point on. And obviously, I don't know if you don't remember Yockey, but he was a staunch atheist and anti-intelligent agency and stuff. But at the same time, he recognized, hey, we have to account for these things. Otherwise, we're not being true researchers and actually following the scientific method based on the evidence. We're just wanting to make as you said, kind of the faith-based illusions, whether it's religion or materialism or something in between. People don't want to. Not a lot of people, I think, don't want to actually address the fundamental necessities of their very existence, which I find very interesting that people want to avoid them.
It might be very easy that there might be a different change, like a different condition where these things like DNA does start just spooling out, and then like it might be that this happened, but the probability is hard to imagine where that is. And certainly on my very little I know about geographic whatever it is, time, but going right back, geological timing,
right,
and what's changed on the Earth. There must be some piece missing from the puzzle.
No, that's very true. Well, Dr. Goult, I greatly appreciate your time. I know we need to cut it off for about 2 hours, so thank you so much for your time, sharing your knowledge with us. I'm really looking forward to November when your next paper comes out, and I'd love to have you back on again to discuss these sorts of things if you're interested, and potentially be able to keep taking advantage, I guess, of your knowledge and your research and all the things you're discovering.
Sure. Thanks for the opportunity and for the good questions. It's been a good discussion. Enjoyed it. But now it's the weekend, so I'm going to say bye.
Absolutely. Have a good one. And folks, thanks so much for joining us, all your support. I greatly appreciate all your super chats and everybody who's been helping to make all this stuff happen. And as I will be having another stream later tonight, and we got another one tomorrow. So make sure to join us. And ultimately, thanks again. And as I always love to say, as you contemplate the underlying necessities of your existence, all the things that you must account for in terms of your understanding of the world, the cosmos, and all the things that just surround us in society, and as you interact with your friends, make sure that the conclusions you reach are logical, plausible, and probable. Have a good one, folks.